everyone. Thank you for joining our webinar today on spinal cord research. I'm Sally Head-Delquist, and I'm president of ImmunoChemistry Technologies. And at ICT, we're on a mission to help cure disease, and we do that by providing better assays to study cell death, which is critically important in spinal cord research, because the sooner the neurons are treated, the sooner they may regenerate. And the biggest goals of early treatment of spinal cord injuries are really to prevent cell death, help those neurons regenerate, and decrease inflammation. And ICT has several assays that can help to measure cell death, and we'll review some of those experiments today. I'm Sally Head-Delquist, and I have a BS in genetics and cell biology and an MBA. I started at ICT by working in the lab, helping to develop these products, and now I'm the president. Assisting me today are Allison Westcott, our customer service manager, and Nick Weninger, our scientific associate, who put together much of this data. So a big thanks to Allison and Nick for helping out today. And to those of you out there watching, we really appreciate you taking the time to make this a part of your day. We've muted your phone. However, we really encourage everyone to ask questions. Just type them into the chat dialog box or send an email to help@immunochemistry.com, and we'll respond directly to you after the webinar is over. And also, we are recording this, and we'll post it on our website and YouTube, so you can watch it later and share it with your colleagues. So watch for an email from Allison, which will contain a link to the recording. I'm really excited today to share some of this work that's really been done on spinal cord injuries. ICT's products have been used by researchers all over the world, and there are a lot of interesting ways they've been used on neurons to look at apoptosis in brain sections and in neurons in vitro, in vivo, and ex vivo. We really want to know what happens to the cells after an injury. Do they live or die? And how can we help them regenerate? I hope this webinar will give you some inspiration when setting up your next experiment. Over 250,000 people each year suffer a spinal cord injury, and it's a life-changing injury. Now it's considered a condition that people can live with, but it's not curable yet. The sooner we can treat the patient, the better the results. Time is of the essence to get this treatment. And after the primary injury, there is a biological cascade of events leading to secondary damage and chronic disease that we'd like to understand. Spinal cord injury directly or indirectly affects many different cell types, including glial cells, smooth muscle cells, and immune cells, as well as connective tissues and blood vessels. So here's a picture of one healthy neuron. On the left is the bulbous perikaryon containing the nucleus with pointy dendrites that receive signals. The cell body stretches into the axon to the right, which branches out at the end and transmits signals through the synapses on the right. Here are two healthy neurons. This is an upper motor neuron and a lower motor neuron that connects to the muscles. The upper neuron initiates the signal in the axon hillock, which goes through the axon into the synapses that contain neurotransmitters that transmit the signal to the receiving neuron, which in turn sends the signal to the muscle and tells it to move. In addition to traumatic injuries, diseases can cause neurological problems too. For example, the axon is insulated by the myelin sheath, which is a lipid layer, and in multiple sclerosis, that's an autoimmune disorder that attacks the myelin sheath, causing nerve damage. When the neural connection is disrupted, these signals are no longer transmitted. The effects can range from mild tingling, like when you bump your elbow, numbness, to complete paralysis where the muscles don't get the message at all. I'd also like to thank the lab of Hiroyuki Kata at the Krembil Research Institute of Toronto Western Hospital for publishing this image in 2019, and a few other pictures that I'll show you today. Their lab really did a lot of great work on spinal cord research. This shows acute damage after a spinal cord injury, and that initial primary injury can cause bones to break, there's internal bleeding, cells die, the neurons stop working, and the normal healing process is disrupted. The blood-spinal cord barrier is disrupted, causing a lot of inflammation and hemorrhaging. Neurons, oligodendrocytes, and astrocytes start dying, and the axons immediately degenerate. and these astrocytes and other glial cells start secreting chemicals such as chondroitin sulfate proteoglycans, which act as a physical barrier, and that impedes normal tissue repair, prevents the axons from sprouting and reorganization of the synapses. And then in addition to the primary injury, that triggers secondary injuries, which can last for several weeks. It's a biological cascade of chemical and physical damage. There's increased permeabilization of the cells, pro-apoptotic signaling, ischemia, lack of oxygen, and molecules are released like increased glutamate and intracellular calcium, which leads to an increase in the reactive oxygen species that damage cellular components like nucleic acids, proteins, and phospholipids. And then the immune system is also triggered. These inflammatory cells infiltrate the spinal cord, including neutrophils, monocytes, macrophages, T cells, and B cells that release inflammatory cytokines such as tumor necrosis factor alpha, interleukin-1 alpha, IL-1 beta, and IL-6. It's a cytokine storm that peaks 6 to 12 hours after injury. There's swelling, scarring, excitotoxicity, reactive oxygen species, immune-mediated toxicity, and just everything is disrupted. There's a lot of immediate damage that happens. But over time, later in the chronic phase, after an injury, a cavity gets formed in the spinal cord, which is surrounded by scar tissue and filled with cerebral spinal fluid. The inflammatory immune cells remain in the area, and these reactive astrocytes change into scar-forming astrocytes, which prevent regenerating axons from crossing the injury. But interestingly enough, lampreys and zebrafish can bridge this gap, and the neurons regenerate. This injury is unrepairable in mammals, but these lower vertebrates can regenerate neurons and fully recover. Can we somehow copy their biology to repair humans? How can we prevent cell death and decrease this inflammation? And to investigate this, it's useful to measure the cell death. And you can easily do that with ICT's products, FLICA and FLIVO. So I'll quickly tell you about them, and then we'll look at the experiments. So FLIVO and FLICA are two methods that can evaluate cell death and apoptosis in vitro, in vivo, and ex vivo. They are labeled peptide inhibitors of caspases that measure apoptosis and pyroptosis. And they're really easy to use. You just add them directly into the tissue section or cell media, or inject them directly into the animal or near the injury, and they bind to active caspases and form a strong covalent bond, and then they're retained within the cell. So they won't react with procaspases nor inactive enzymes. And the reaction is fast. It starts happening with 15 minutes of adding to the sample, and they're non-cytotoxic with minimal background staining, and they'll remain inside the cell for detection as long as the cell membrane is intact. And they're compatible with other stains like GFP, DAPI, and Hooks. They can be detected at many wavelengths like red, green, near infrared, and at 660 nanometers. So you can detect them with a microscope, plate reader, flow cytometer, a whole animal imager, and other equipment. They're available to study a variety of different caspases, and which reagent you use for your experiment really all depends on which instrument you're going to use for analysis, what other stains you're going to use with it, and which caspase you want to target. Of course, all of these kits are available online, so you can order them anytime. Now, let's show you how these will work in some spinal cord studies, and I'll start by showing you the lamprey brain as a model system. So sea lampreys have the amazing ability to completely recover from a spinal cord injury. Lampreys existed before the dinosaurs and have not really changed in the last 360 million years. They're living fossils, and they're one of the most valuable animal models for studying vertebrate brain development because they have just one brain and just one spinal cord. The nervous system, neural synapses, and the synaptic neurotransmitters are really the most primitive state in vertebrates. And they have relatively few nerve cells, and it's a simple neural network that's easy to study. They also have many visible neurons, and they survive for a long time in vitro. The lamprey brainstem has about 30 large identifiable descending reticulospinal neurons, and here's a schematic of this simple neuronal network. This is the dorsal view of the back, and these neurons are in pairs, so there's 2 in each spot. And all of them can be labeled by FLICA and FLIVO. This drawing is from the lab of Anton Barrero Iglesias at Shriners Pediatric Research Hospital and Temple University, as well as the University of Santiago in Spain. And this group published many papers investigating these neurons, so we'll take a look at a lot of their experiments. So thanks to them for all their great work. These projecting neurons of the brainstem really vary greatly in their ability to survive, despite the fact they have analogous projection pathways. Some of these neurons are considered good regenerators, meaning they regenerate their axon more than 55% of the time, and others are considered bad regenerators, meaning that they regenerate their axons less than 50% of the time. And they're frequently the targets of many studies. One of the bad regenerators I want to point out is the MTH at the bottom. That's the Meissner neuron. And we'll look at a couple of experiments looking at that neuron. In this experiment, they wanted to visualize and directly label apoptotic neurons in whole-mounted brains after a complete spinal cord transection. And this was easy to do using FLICA. The FLICA is cell-permeant, so you just add it to the media for an hour. It covalently binds with active caspases, and the signal is retained in the neuron. The brains were then washed 9 times and fixed with 4% paraformaldehyde overnight and covers slipped with Prolong. These photos are 10x, and they used the FLICA reagent in green because they were looking at all caspase activity using the polycaspases reagent FAM-VAD-FMK. And they successfully detected caspase activity in this whole-mounted brain without using antibodies. This publication was a precursor to many other experiments, and it really has become a widely accepted method and is even chapter 23 of this methods book called Neuronal Cell Death by Humana Press. And the Meissner neuron here, you can see the MTH. It turned green. It's apoptotic on the right, which was 2 weeks after the spinal cord injury, but it's not green in the control animal on the left in B. Here is another view of the Meissner neuron. This is a dorsal view of a whole mount. Rostral is to the right. That's toward the head of the animal. And the top photo shows the site of injury, while the bottom picture shows the area midway between the injury and the brain. And we can see that 2 hours after the injury, caspase 8 is activated. It's green in the top picture, but not midway. It's dark in the bottom picture. That means that apoptosis is activated via the extrinsic pathway. Caspase 8 is present in neurons considered to be bad regenerators, and this fundamental work was frequently cited in other studies as a really convenient vertebrate model to study retrograde degeneration of descending pathways after a spinal cord injury. Here's an overview of this basic procedure that's been used in many studies. They cut the spinal cord. In some animals, they applied DTMR to label regenerating neurons or a morpholino to the site of the transection at the time. Then they let the animal recover for 2 weeks and then dissected the brains. And once the brains were dissected, they added FLIVO for an hour or so, washed them, then fixed in paraformaldehyde, and then in 70% ethanol, ETOH, and then stored the brain sections frozen at -20. And the FLICA signal remained strong throughout this processing and was compatible with many other procedures that they did after FLICA labeling. It's important to note that they stained the sections with FLICA first, and then they fixed them. In this paper, published by researchers at the Shriners Hospital, they wanted to identify the early stages of apoptosis after injury. Time really is of the essence after an injury, and they found that there is a delay in the process of cell death, which could provide time to treat the injury by preventing cell death and limiting secondary damage, inflammation, and by encouraging regeneration. So they were looking at the neurons considered bad regenerators. In this one paper, they did a lot of experiments I'd like to highlight. They were looking at caspase activity as well as other biomarkers using antibodies, immunohistochemistry, in situ hybridization, tunnel, DTMR, and antisense morpholino probes for mRNA. This is from figure 5 of this paper where they were looking at tunnel. They were comparing FLICA with tunnel. And tunnel is a widely used method to assess apoptosis in tissue sections by detecting DNA breakage. But in lampreys, in the spinal projecting neurons, they show this delayed cell death after transaction. So tunnel can't be detected until 4 weeks after injury, and it's a more complex procedure. So therefore, this experiment was done 24 weeks after an injury, whereas most of the experiments were done 2 weeks after an injury. And this shows that FLICA and tunnel show 100% co-localization in the tissue sections. So FLICA and FLIVO are a fast, easy, and early reliable method to detect apoptosis in the Lamprey nervous system. Here, the researchers wanted to confirm the specificity of FLICA by pre-incubating some control animals with the unlabeled inhibitor Z-VAD-FMK. And this shows that Z-VAD-FMK significantly reduced the green FLICA staining in the picture D on the lower right compared to C on the left. And this is quantified in the graph in E, meaning that FLICA is acceptable to label caspase activity in lamprey. Here they also stained with an antibody. This is a whole-mounted brain from a post-metamorphic adult lamprey 2 weeks after a spinal cord transection. The brains were labeled with FLICA, which is green, and then fixed and frozen. Then they were labeled with an antibody for neurofilaments, that's LCM16, and then visualized using goat anti-mouse HRP with a DAB substrate. The M2 neuron in D is labeled with the antibody, but it is not green in C at left, so it's not apoptotic. And there's a bigger picture of it in G. In this experiment, they wanted to detect neurons with caspase activity in PTP sigma. This is a larval lamprey, and like before, the brain was transected first, and then 2 weeks later it was dissected and then labeled with FLICA fixed and frozen. Then they performed in situ hybridization to find mRNA for PTP sigma. And yes, this experiment worked really well and showed excellent staining. And it showed that the mRNA is preserved well enough during the FLICA treatment and processing to successfully perform in situ hybridization. In this experiment, they wanted to detect neurons with caspase activity and looked at retrograde degeneration with DTMR, which is red, and they wanted to see if FLICA would influence the growth pattern of the neurons, and it did not. So this experiment was done a little differently than the previous methods. So when the spinal cord was transected, it was also labeled with DTMR at the time of injury, and that was just applied to the site of the injury. And then the animal was released for 2 weeks and allowed to recover. So after 2 weeks, then the brain was dissected, prepared, and labeled with FLICA and fixed. So this shows that this DTMR and FLICA are compatible techniques. This data is from larval C lampreys 2 weeks after a spinal cord injury. The brighter the FLICA signal, the less likely the neuron is to regenerate, such as neuron I1, which is the brightest yet least likely to regenerate. We know that there's a slow process of cell death for weeks after a complete spinal cord transection, with secondary injuries and chronic damage long term. If we can understand when and how caspases are activated, that may lead to treatments for spinal cord injuries. How do we keep these cells from becoming apoptotic, and when can we best do that? Looking at apoptosis using FAM-VAD-FMK for caspase 8, we see that it's the extrinsic pathway that mediates cell death. So caspase 8 carried by microtubules to other areas and is inhibited by treatment with a drug called Taxol, which we see in the bottom. So the top picture is the normal control showing typical cell death and apoptosis in the green neurons versus in the bottom after treatment with Taxol, that apoptosis is inhibited, so it's not green. More about Taxol as a treatment, it helps survival of the neurons, and Caspase 8 signaling seems to be transported by microtubules from the site of the injury to the neuronal soma. And so preventing activation of this process could be an important therapeutic approach after a spinal cord injury in mammals. And so they find that Taxol inhibits the transport of the signal that activate Caspase 8, but it did not prevent the initial activation of Caspase 8. Using the FLICA reagent in red, that's the sulforhodamine polycaspase FLICA reagent, it predicted regeneration of the neuron. So here the control brains showed activated caspases in very few neurons. Moreover, when they looked at it at 1 week, 2 weeks, 4 weeks, and 8 weeks after the transection, the probability of caspase labeling in individual identified neurons correlated inversely with the probability of axon regeneration. So the inverse correlation was statistically significant at all time points and suggests that as an apoptosis signaling indicator, FLICO can also help predict whether individual neurons will undergo axon regeneration after an injury. In this experiment, they were also looking at a molecule called RhoA, and they know that RhoA expression levels in the brain are enhanced after spinal cord transection, and the numbers of FLICA identified positive neurons in the brain were also greatly increased. Therefore, would an in vivo knockdown of RhoA protect these neurons from dying and promote axon regeneration? And yes, it did. So let's take a look at that data on the next slide. So RhoA could be used as another avenue of treatment. So again, these are researchers from the Shriners Hospitals, and they were looking at RhoA, which is a small GTPase that inhibited retrograde apoptosis after spinal cord injury. And RhoA can be activated by the chondroitin sulfate proteoglycans, MAG, and NOGO. Here they use the red FLICA product, SR-VAD-FMK, that's sulforhodamine, along with a morpholino that was tagged with green fluorescence. And a morpholino is a type of oligomer used in molecular biology to modify gene expression and antisense oligonucleotides, those MOs. So the control MO should not interfere while the experimental MO blocks the action of RhoA. And as expected, if RhoA is blocked, apoptosis decreases. The red fluorescence drops, which you see in the bottom row. And the MO was applied at the time of transection, like the DTMR. Then the animals recovered for 2 weeks. Then the brain was dissected and SR FLICA was applied and fixed and imaged. So the RhoA knockdown significantly reduced this FLICA labeling and was especially pronounced in reticulospinal neurons that are poor regenerators. So this suggests that RhoA is involved in retrograde neuronal cell death after a spinal cord injury, and that the RhoA knockdown is beneficial for neuronal survival after axotomy. In this experiment, they wanted to see if sealing the neuron with PEG, that's polyethylene glycol, would help it survive, and it did. These animals were transected at 20% of their body length and treated with or without PEG. And 24 hours later, they applied DTMR, which is red, and let the animal recover for 2 weeks. And after those 2 weeks, they dissected the brain, labeled with FLICA in green, and fixed them. So the animals treated with PEG showed a significant reduction in the uptake of DTMR and reduced caspase activity, which means there's less green above in figure F. They also did an experiment to see if calcium ions would make a difference, but it did not. The axons were still apoptotic. So here's a graph of what they think happens when the axon is cut. The cytosolic calcium increases, which injures the mitochondria, which release more cytochrome C, and that activates the intrinsic apoptosis via caspase activity, causing more and more cell death. However, quickly resealing the axon with PEG helps it survive. So that's a good strategy to pursue for treatment. Reticular spinal neurons in lamprey project almost the entire length of the animal. And in this paper, they wanted to know, does it make a difference where the injury is? Will it still regenerate? And yes, there was an inverse relationship between the reticular spinal neuron size and the probability that the axon would regenerate and that the neuron would undergo delayed apoptosis. And they did this study using FLICA. So the bad regenerator neurons are typically larger, including the caliber axons, which take longer to reseal. And at any transaction level, as percentage of body length, the larger the neuron and its axon, the longer it takes for its axon to seal. And the longer it takes to seal, the more likely the perikaryon will undergo delayed retrograde cell death. Once we've identified the neurons, how can we protect them? One finding is that GABA is massively released after a spinal cord injury. And here these researchers found that GABA, GBAB, and baclofen reduce apoptosis in the neurons, and signaling through these receptors promotes survival and regeneration of descending neurons. And one potential treatment for protecting severed neurons is mucimol. And mucimol is a GABAA receptor agonist, and treatment with mucimol decreases apoptosis in descending neurons. And these pictures are whole-mounted brains with a control on the left, or mucimol on the right, which shows a significant reduction in green fluorescence. So you know there's less apoptosis occurring. This is the last slide we'll look at on lampreys. And here they were studying a molecule called neogenin and did a time course study from 1 to 16 weeks. So in addition to staining caspases, they also used an in situ probe for neogenin mRNA, and they also studied the swimming behavior to assess recovery, which I showed here on the right. So the circles on the right are the typical examples of a swimming path of one animal. And at 16 weeks, you see on the bottom, the lampreys treated with MIS-MO and then neogenin MO swam identically to the control uninjured lampreys, which you see the circle in the upper left. A little bit about zebrafish. They are really even more amazing than lampreys. They're a favorite model to study regeneration and seem to have unlimited regeneration abilities. They've even been known to regrow in their tail fin 27 times, and they can make a complete recovery after a spinal cord injury and regain all locomotor function. And unlike mammals, zebrafish use these glial cells to help bridge the spinal cord. They help repair the spinal cord injury, and then they use another molecule called connective tissue growth factor A, that's CTGFA, to activate regeneration. And humans also have CTGFA, but we can't regenerate. So this is something to be investigated further. You know, this experiment using zebrafish is really amazing for many reasons. They were inspired by all the experiments done on lampreys and used the FLICA with zebrafish. They used a double transgenic fish to look at neuroregeneration and the immune response to see if it's intrinsic or extrinsic. And these were larval zebrafish that expressed red M cherry in the motor neurons, pancreas, and heart, as shown here in this picture. You can see them brightly on the picture. And the immune system probably plays an important role in regeneration. For example, there's a strong microglia and macrophage reaction to a spinal lesion, and this activation of microglia and macrophages alone is sufficient to trigger neuronal regeneration in adult zebrafish telencephalon. So that means that there's some sort of signaling of microglia and macrophages to progenitor cells. In the FLICA experiment, the zebrafish were treated 72 to 78 hours post-fertilization, either with a control, DMSO, or with a drug MTZ. Then they added FLICA or FLIVO directly to the water, which entered the animals and successfully labeled apoptotic neurons green, which you see on the bottom. Then they fixed the animals and mounted them in glycerol, all while preserving the signals. They also did some tunnel experiments. They found that MTZ leads to ablation of spinal motor neurons and heart cells in these transgenic larvae. MTZ treatment actually leads to ablation of all transgene expressing cells and to microglia and macrophage activation, which in turn leads to caspase activation and apoptosis, which you see is the green stain in the bottom on the left. The cells are also expressing the red M cherry. Researchers at the Karolinska Institute in Stockholm, Sweden, developed a consistent way to create a spinal cord injury in zebrafish. And to develop this method, they used HeLa cells, which you can see cultured in the bottom left. They perfected the technique, and they used the green FAM-FLICA to measure cell death. Those are the black arrows, are the apoptotic cells in G, so they could specifically identify which cells are being killed by the laser. And I thought this was a pretty neat technique. They targeted the entire 10th segment of the spinal cord, and this method is a more precise way of generating a spinal cord injury, and it has this single cell resolution. So this will enable researchers to study a range of injuries from minor damage to complete transection. So we look forward to seeing more studies in zebrafish. Finally, we'll look at one more paper using mice, which are mammals and have similar nervous systems as humans. So again, this is researchers at the Karolinska Institute in Stockholm, Sweden. They used ICT's green FAM-FLICA kit for Caspase 1, which is FAM-VAD-FMK, to assess pyroptosis in mice neurons. In this study, they were investigating why ischemia reperfusion, that's a lack of oxygen, causes death in spinal neurons. They found that if they inhibit activation of the AIM2 inflammasome, that will restore the motor function. So let's look at their data. The experimental animals had this restricted blood flow to prevent oxygenation of the tissues, and then the blood flow was restored, causing this ischemia reperfusion injury. And all the injured mice lost motor function. Then their cerebrospinal fluid was collected and added to cultured neurons, and they were labeled with the FAM-VAD-FMK. So this shows elevated caspase 1 activity and pyroptosis in the challenged cells on the bottom. They also stained with an antibody and DAPI, which is a blue DNA stain. Lastly, I wanted to leave you with some final thoughts about what does this mean for people. There are more and better treatments on the horizon, including stem cell transplants. And I thought this was a really nice table of some of the important research that's going on today. There's a lot of progressive work being done with different drugs and these stem cells, which really makes me optimistic that someday we'll be able to fully repair spinal cord injuries. And remember, we've recorded this webinar and we'll be sending you a copy of it, so you'll be able to look back and review and read all these slides in more detail. So this wraps up our presentation for today. If you have any questions, feel free to send them through the chat box, and we'll respond directly to you or email them to help@immunochemistry.com. Of course, you can always call us. We love hearing how you are using our products and helping you set up your experiments. FLICA and FLIVO are two methods that are easy, fast, and reliable that can be used to evaluate cell death in spinal cord and neuronal studies in vitro, in vivo, and ex vivo, and they're available for you to order online through our website. So again, thanks for tuning in today and for all the research you're doing. And thanks again to Allison Westcott, Nick Wenninger, and Kristen Pauly, our marketing team, for managing these webinars. We here at ICT are really proud to be part of the medical research community and provide tools like FLICA and FLIVO that can help understand and someday cure spinal cord injuries. I'm Sally Head-Dahlquist, President of ImmunoChemistry Technologies, and it's been a pleasure helping you find a cure.